Well, thank you very much for giving me the opportunity to present this work, and I do have to apologize that some of the slides were presented at ACC very recently, but I have added new data into this talk. So I'm going to talk about our use of mate pair sequencing to characterize chromosomal rearrangements in AML. I have no disclosures. So as we know, acute leukemias are rapidly progressing malignancies of early differentiated cells. They could be of myeloid, lymphoid, or mixed lineage. Today I'm going to talk about AML specifically. This is the most common form of acute leukemia amongst adults. It's characterized by a clonal expansion of myeloid blasts, and the SEER database indicates that most likely we will have 20,000 new cases of AML in 2018 and approximately 10,000 deaths. The median age at diagnosis is 67 years, but as we know, it can also affect the pediatric population. AML diagnosis and prognostic assessment incorporates a variety of different factors, including clinical metrics, morphology requiring at least 20% bone marrow blasts, with the exception of the core binding factor types 821, inversion 16, and even 1517. Immunophenotyping, molecular genetics for looking for abnormalities in genes such as NPM1, FLT3, CBPA, RUNX1, TP53, and so on. And obviously cytogenetics, which plays an important role in predicting overall survival. Those with favorable cytogenetics include those core binding factor 1517 also, and adverse outcomes are found in those with KMT2A rearrangements, 922, inversion 3, deletions of chromosomes 5 and 7, and so on. So in the evaluation of a case of suspected AML, the ASHCAP guidelines strongly recommend a conventional chromosome analysis, and it is considered mandatory in the ELN recommendations. However, recent studies have highlighted the potential of novel sequencing technologies. And so we specifically ask, can our mate pair sequencing test replace chromosomes in FISH on diagnostic AML cases? So I'm going to spend the next 2 slides giving a brief overview of mate pair sequencing. It is an NGS-based method that allows for the efficient detection of structural variation, including balanced and unbalanced translocations and inversions, as well as copy number variation. So in the next slide, I'll talk about our utilization of a specialized library preparation, followed by whole genome paired-end sequencing. And within a single test of mate pair sequencing, we can identify all abnormalities provided by chromosomes, FISH, and microarray. It is important to note that the limitations are we cannot detect single nucleotide variants, and also it's not useful for MRD detection at this time. So just briefly, we utilize the Nextera Illumina mate pair library preparation, which generates fragments that are 2 to 5 kb. These are labeled with biotin molecules. The fragments are circularized to bring those ends together, and then we undergo a second fragmentation step, giving rise to 200 to 500 base pair fragments. We do paired end, we pull down these fragments using streptavidin and do paired end sequencing. So when we run the sequencing results through our algorithm, we're essentially hunting for discordant fragments. So when we have fragments that are mapping farther away, then this gives us information that there could be a duplication. When we map closer together, a deletion, or if we have an unexpected orientation, this could be a sign of an inversion or unexpected chromosome location, fragments on different chromosomes, then we have a translocation. So this test is live, and what we are doing now is developing panels so that ultimately we can replace our FISH assays. And so this specific test that we validated is for AML. And while the mate pair sequencing test is genome-wide, we don't actually at this time want to see all abnormalities in the genome. We are specifically looking for those that are unique to AML, specifically on our AML FISH panel. And so we utilize a software that was developed at Mayo called Ingenium, where we can have filters, and we're only looking for those abnormalities that are on our FISH panel. So on the upper right corner, you see a genome plot, which basically identifies all the chromosomes, and you can appreciate deletions, duplications, as well as translocations, which are visualized by the black line. Junction plot, where you can specifically zoom in and identify a chromosomal rearrangement, and in this case, this is a BCR-ABL translocation. When we performed our validation study, we utilized 118 specimens. Our pre-validation work, this is important to note, we did observe that when we had less than 25% abnormal clone, we were at risk of missing copy number changes. So we require at least 25% of our clonal fraction to be abnormal, and fusions, we require at least 10% of our fraction to be the abnormal clone. When we compared 68 known abnormal AML samples based on FISH to mate pair sequencing, in the detection of the primary abnormality, we had 100% sensitivity. We looked at 20 karyotypically normal AML samples as well as normal non-AML samples, and we did not identify AML abnormalities, achieving 100% specificity. So the next two slides, I want to highlight some of the value of mate pair sequencing. In this specific case, we had a 5-year-old female with a diagnosis of AML. Chromosome studies were normal, and our KMT2A break-apart FISH had a normal pattern. However, mate pair sequencing did identify an insertion of exons 9 to 24 of the MLT10 gene into intron 9 of KMT2A. Then when we did our MLT10 KMT2A dual color dual fusion probe, we did identify an abnormality, but this was missed on the clinical workflow. So MatePairSeq was able to identify this cryptic insertional event that was missed using a KMT2A break-apart FISH probe. In addition, MatePair sequencing identified novel fusions in 8 abnormal cases where we did identify an abnormal FISH pattern. So the case on the bottom shows that while this patient did not have a BCR-ABL rearrangement, we did identify 3 BCR signals, and MatePair sequencing proved an FGFR1 BCR rearrangement, giving this patient the WHO myeloid neoplasm with FGFR1 interpretation and the potential for FGFR1 inhibitor treatment. We have a NUP98 break-apart FISH probe. However, MatePair sequencing was necessary to fully characterize the partner gene for those NUP98 cases, and we had 2 that were NUP98 KDM5A for acute megakaryoblast leukemia. So while we validated this test, so we have done a technical validation, what we really wanted to understand was what's the clinical utility of this test. And so we are in the process of doing this study now. Our target is 100 patients, and we've analyzed 64 so far. So essentially what we're doing is taking all of Mayo Clinic newly diagnosed AML cases. The physician is ordering chromosomes and FISH, and we, in the background, are performing MatePair sequencing on all cases, as well as in some, also performing RNA sequencing. We have, as you can appreciate, we have 64 cases. The majority of these are adults with a median age of 68 years with a range of 2 to 90, and the mean blast percentage was 53%. So this is demonstrating the types of classic cytogenetic abnormalities that we identify based on FISH and chromosome studies. The majority of these are representing normal karyotype or abnormalities involving chromosomes 5 and 7 in 62% of cases. So of these 64 cases, we compared what we found by MatePair sequencing and what we found by chromosomes and FISH. And we were able to identify all known primary abnormalities in 63 out of 64 cases, achieving a 98% sensitivity. MatePair-Seq did miss one abnormality in a case with a deletion of chromosome 11, and this was not identified by MatePair. What was also interesting was when the physician only ordered chromosomes and not FISH, we were actually able to identify abnormalities on MPseq that were not appreciated on chromosomes. And on the bottom indicates a case that was interpreted as a deletion of 7q, but in fact, there was an inversion 7 and an MLLT10 KMT2A rearrangement that would have been picked up had the clinician ordered the FISH assay, but they only ordered chromosomes. Another case was interpreted as AML with trisomy 8, but we identified a cryptic NUB98 NSD1 rearrangement that would have been picked up had the clinician ordered the NUB98 break-apart FISH. Also, we proved that this was a functional fusion by RNA sequencing. So in 42% of cases, MPseq identified additional AML abnormalities in addition to the primary abnormality. And when I say AML abnormalities, we have an AML list that we utilize that was essentially put together by Beth Patel, and we're looking for any gene that has ever been associated with an AML. So there are additional abnormalities, but this is a way that we can at least look for those that are associated with AML. And in 53% of cases, we did not identify any additional AML abnormalities. And so the majority of the normal karyotype cases fall within this category, versus those cases that have a deletion 5 or deletion chromosome 7 have additional AML abnormalities. So we wanted to look at these cases a little bit further. And what you can appreciate on the upper part of this figure is that those cases that are with normal karyotype AML have a relatively quiet genome. And those, as we all know, are often characterized by point mutations, and we, in fact, do see that based on our clinical grade NGS assay. So those cases that have a deletion of chromosome 5 or 7 have a very complex genome, as you can appreciate, with multiple rearrangements found. And in these cases, the majority of these had TP53 mutations. So we wanted to further quantify how noisy the genome was. And so Beth painstakingly added all of the variants, and these were identified based on if there was an insertion or inversion, a translocation, or copy number gain or copy number loss. And what you can appreciate is that those with co-deletion of 5q and 7q have the most complex genomes with an average of 108 variants per case versus those that have a monosomy 7 or a deletion of 7q with only 16 events per case. So in conclusion, when we underwent a technical validation of this assay, we achieved 100% sensitivity and specificity in the detection of the primary cytogenetic abnormality. This test is suitable for diagnostic AML cases, but not for follow-up cases, which is very important to note. We are working on improving the algorithm to have a better sensitivity. MPseq can identify cytogenetically cryptic rearrangements or clarify abnormal chromosome results. We can identify specific AML gene fusions in 12% of cases when we did identify an abnormality by FISH. MPseq identified additional over and above FISH and chromosomes for genome-wide AML-related abnormalities in 42% of cases. And most normal karyotype 15, 17, and inversion 16 cases had no additional AML-specific abnormalities. And cases with deletion 5, 7, or monosomy 7 had the most complex genomes. So MPseq does provide an alternative technology to comprehensively evaluate for chromosome rearrangements and copy number changes in a single assay rather than large panels of independent FISH probes or a chromosome study. So finally, I want to acknowledge our huge group, and also specifically Patty Gripe, who has played an important role in this project, as well as Umut Apar, who is now at Sloan Kettering, and Nicole Hotman, and also mainly Beth Patel, who has put a lot of effort into this project as well. So finally, thank you very much, and I can answer any questions. And this is actually our RNA sequencing result that I didn't really spend a lot of time talking about, but we're happy that we can at least get this to work. So.